Good afternoon, everybody. I am Caroline Dive, and it's my pleasure to be the EACR President-elect. Welcome to the third session of EACR 2020. We've had some amazing science. It is now my absolute honor to welcome and introduce Nobel laureate Professor Bill Kaelin as the first speaker of the session. Bill is, of course, world-renowned for his outstanding research on tumor suppressor proteins, notably RB, von Hippel-Lindau, and P73. Bill has dedicated his research efforts to developing therapeutic strategies based on the detailed molecular knowledge of tumor suppressor protein functions. He received his MD from Duke University, trained in internal medicine at Johns Hopkins, and was a clinical fellow at Dana-Farber, and whilst there, postdoc with David Livingstone, where his love affair with tumor suppressors began. Bill started his independent laboratory at the Dana-Farber, became a Howard Hughes investigator, and is currently professor of medicine at the Harvard Medical School. Bill is a member of the AACR Academy and a fellow of the National Academy of Sciences, and he's amassed more prizes and awards than I could possibly tell you about right now, but they include the ASCO Science in Oncology Award and the Lasker Award for Basic Medical Research. I'm sure you all know that Bill was awarded the Nobel Prize in Medicine and Physiology jointly with Peter Ratcliffe and Greg Semenza for the discoveries of how cells sense and adapt to oxygen availability. Bill's talks are legendary and they're always thought-provoking. So I'm delighted to announce his EACR 2020 keynote entitled Von Hippel-Lindau Disease. Welcome, Bill.
Good afternoon. It's a pleasure to be with you, at least virtually today. Here's my disclosure slide. The most relevant disclosure is my involvement with a company called FibroGen, and I'll refer to that later. So here are the people who've worked on von Hippel-Lindau disease over the years in my laboratory, and I'll try to mention specific individuals as I go along. And we've also had many very talented, very generous collaborators. I can't possibly list them all, but here are some of the individuals who helped us with the work that led to the Nobel Prize. So when I was younger, I was convinced I was going to be a clinical doctor. Here's a picture of me as an intern in internal medicine at the Johns Hopkins Hospital. And while I was an intern, my chairman was the great Victor McKusick, shown here. And Victor was both a medical geneticist and a medical historian. And so I like to start this story on one level where it began, which is with this paper by Treacher Collins that appeared in 1894. Treacher Collins had seen a brother and a sister, both of whom had an unusual retinal lesion that we would now refer to as a retinal angioma or a retinal hemangioblastoma. These are vascular neoplasms of the retina. They're easiest to see if the patient is first injected with a fluorescein dye, such as has been done here. And you can see this patient's large retinal hemangioblastoma. A similar family was then described by Eugen von Hippel. He likewise identified a family where multiple individuals had retinal hemangioblastomas. But the real hero in this story, by all accounts, is the Swedish neuropathologist Arvid Lindau. Lindau, being a neuropathologist, appreciated that these retinal lesions were often associated with other tumors, and in particular were associated with hemangioblastomas of the cerebellum and the spinal cord. So I show you here what some of these lesions look like. So in the upper left, I'm showing you the fundoscopic view of a patient with a retinal hemangioblastoma. Here's the tumor shown here for orientation. This is the normal optic nerve. I've already mentioned that these lesions are easiest to see if the patient is first injected with a fluorescein dye. So here's a VHL patient who has multiple small retinal hemangioblastomas. But as I alluded to a moment ago, these retinal hemangioblastomas are sometimes associated with hemangioblastomas of the cerebellum or spinal cord. So here on the lower left, we have a sagittal MRI of a patient with VHL disease. For orientation, here's the normal lateral ventricle. But this patient has a large cystic hemangioblastoma with the solid component enhancing here. by MRI arising in the cerebellum. Now, in addition, these patients can also develop renal cysts, and over time, these renal cysts can develop into renal cell carcinomas. So here we're looking at a renal cyst in the kidney of a patient with VHL disease. The urologist has unroofed this cyst and has discovered a clear cell renal carcinoma, which is the most common form of kidney cancer, arising from the epithelial cells lining the cyst cavity. So this came to be known as von Hippel-Lindau disease, with apologies to Treacher Collins. It affects about 1 in 35,000 people. In hindsight, there are some families that have been affected with this disease dating back to at least the 17th century. Clinically, it appears as an autosomal dominant condition, although, as you'll see in a moment, it's actually caused by inactivating mutations of a tumor suppressor gene. We've talked about the central nervous system and retinal hemangioblastomas. I mentioned that these patients can also develop clear cell renal cell carcinoma, which is the most common form of kidney cancer. They can also develop so-called paragangliomas, and when these paragangliomas arise in the adrenal gland, they're referred to as pheochromocytomas. Now, I've highlighted in red clear cell renal cell carcinoma because hemangioblastomas and pheochromocytomas are fascinating, but they're admittedly relatively rare, whereas clear cell renal cell carcinoma, again, is the most common form of kidney cancer, and kidney cancer is one of the 10 most common cancers in the developed world. So shortly after I started my own laboratory in 1993, the VHL gene was isolated by a group led by Marston Linehan, Bert Savar, and Michael Lerman at the National Cancer Institute, working with Eamon Mayer, who at that time was in the University of Birmingham in England. Now, it turns out on further inspection that VHL disease is caused by inactivating mutations of the VHL tumor suppressor gene. So patients with VHL disease have inherited a defective version of the VHL gene from mom or dad. So here I'm showing you the maternal and the paternal copies of chromosome 3 schematically. The VHL gene lives on the short arm. And this schematic, the individual has inherited a defective version of the VHL gene from mom. But initially they're okay because they still have a remaining wild type allele. But if a susceptible cell in the brain or the kidney spontaneously loses the remaining wild type allele, that cell can go on to form a tumor. So this looks like a so-called Knudsen two-hit tumor suppressor. And as you would predict from the knowledge that germline VHL mutations predispose to, for example, clear cell renal cell carcinoma, if you now look at non-hereditary or sporadic clear cell renal cell carcinomas, you again see that these tumors frequently have mutated or lost both the maternal and the paternal VHL alleles. But here, both hits, if you will, have occurred somatically, in contrast to VHL disease, where the first hit has occurred in the germline. Now, again, when I was younger, I thought I was going to be a doctor. In fact, here I am in 1987 as a chief resident. So I knew a lot about the clinical features of diseases like VHL disease. And so, for example, I knew that the tumors that develop in VHL disease are highly angiogenic. We've already talked about the retinal hemangioblastomas, but it's also true that kidney cancers are notoriously rich in blood vessels. And in fact, in the pre-CAT scan era, the diagnostic procedure of choice in patients suspected of having a kidney cancer was to do a renal angiogram and to look at the characteristic appearance of new blood vessels. And I'll remind you that in the 90s, there was intense interest in developing angiogenesis inhibitors as a way of treating cancer. And so it seemed to me that studying the VHL gene, in addition to teaching us about cancers like kidney cancer, might also teach us about the control of angiogenesis. Now, another curious feature of the tumors seen in VHL disease is that they sometimes cause patients to make too many red blood cells, which is the opposite of anemia, where you have too few red blood cells. So normally, if you were to take a patient's blood and spin down the blood in a centrifuge tube, about 50% of the volume or 40% of the volume would be made up of red blood cells. But in patients with VHL disease and VHL-associated tumors, sometimes they make too many red blood cells and have an increase in the packed red blood cell volume, the so-called hematocrit. Now, here's sort of a laundry list of the diseases and conditions that are known to cause excess red blood cell production. Some of these are clearly adaptive, such as in certain individuals who live at high altitude or who have chronic lung or heart conditions where they're not delivering enough oxygen. But again, it was known that a number of tumors can cause excess red blood cell production. And it had always struck me as odd that the 3 tumors that are seen in VHL disease make this list, especially given the fact that cerebellar hemangioblastomas and pheochromocytomas are otherwise fairly rare. So taking this all together, these VHL-associated tumors occasionally stimulate new blood vessels to form. They stimulate angiogenesis, and that's because they can produce the angiogenic growth factor, VEGF. And I just told you these VHL-associated tumors can sometimes stimulate red blood cell production, and that's because they can ectopically produce erythropoietin. And what VEGF and erythropoietin have in common is that they're normally induced when cells or tissues are not getting enough oxygen. And so that suggested to me that there must be some link between the VHL protein and how cells sense and respond to changes in oxygen. So to develop, hopefully, a tractable cell-based model to study this, we performed northern blots, and this was work done by Othon Iliopoulos with the help of Mark Goldberg and Andrew Levy. What was done here was to do northern blots to measure the abundance of the VEGF mRNA, as well as 2 other well-studied hypoxia-inducible mRNAs, the mRNA for PDGFB and the mRNA for GLUT1. So the cells in this experiment were grown either under 1% or low oxygen conditions, or under 21% or high oxygen conditions. And so let's begin by looking at HEP3B cells. So HEP3B cells are VHL proficient. They were a workhorse cell line used to study so-called hypoxia-inducible mRNAs. And in these cells, as expected, these hypoxia-inducible mRNAs only accumulated if the cells were grown under low oxygen conditions. In contrast, 7860 cells are a renocarcinoma cell line that lack an intact version of the VHL gene. And in these cells, we observed high levels of these hypoxia-inducible mRNAs, whether oxygen was present or not. Othon Iliopoulos then took these cells and by stable transfection restored the function of the VHL protein. So here are 3 independent subclones where he restored the function of the VHL protein, and you can see that he restored the regulation of these hypoxia-inducible mRNAs. And this was specific because this was not observed if he instead stably transfected the cells with an empty vector or with a vector encoding a tumor-associated VHL mutant that's shown here. And so summarizing this experiment, this said that loss of the VHL protein really uncoupled the accumulation of these hypoxia-inducible mRNAs from actual oxygen availability. So in the VHL proficient cells, these hypoxia-inducible mRNAs only accumulated to high levels under low oxygen conditions as expected. But in the VHL defective cells, these hypoxia-inducible mRNAs accumulated at high levels irrespective of changes in oxygen availability. So in parallel, we did biochemical experiments to look for VHL-associated proteins. And what I haven't told you so far is that the primary sequence of the VHL protein didn't really provide any clues as to its possible functions. And in particular, the VHL protein wasn't similar to any other known proteins. But Adam Keibel, Kim Lonergan, and others in my laboratory showed that the VHL protein forms a stable complex with a long in C, a long in B, and Col2. Moreover, a region of the VHL protein that's frequently mutated in VHL disease, which is now referred to as the alpha domain, was shown to be necessary and sufficient to recruit these other binding partners. Now, this turned out to be a big break in the story because Steve Elledge, who was then at Baylor but is now at Harvard, pointed out to me that a long in C resembles a yeast protein called SCIP1, and Col2, a member of the Cullen family, resembles a yeast protein called CDC53. And these 2 proteins, when bound to a so-called F-box protein, generate a ubiquitin ligase complex that's capable of targeting other proteins for degradation. And so purely by analogy, we began to think that maybe the VHL protein served the same role as an F-box protein. This idea was strengthened further by structural studies we did in collaboration with Nikola Pavletić, which further underscored the potential similarity between VHL and FBOX proteins, and moreover, identified a second hotspot for mutations, which we now refer to as the beta domain, which we predicted would be the substrate docking site for this putative ligase. Now, many of these so-called hypoxia-inducible mRNAs are under the control of a transcription factor, which is called hypoxia-inducible factor, or HIF. It contains an unstable alpha subunit that's normally degraded in the presence of oxygen, as well as a constitutively stable partner protein, which is variably called HIF beta or ARNT. And this transcription factor had been very well studied by a number of laboratories, including the laboratories of Greg Semenza, Peter Ratcliffe, Frank Bunn, Jaime Caro, and many others. Now, a big break in the story came when Peter Ratcliffe's group showed that in cells lacking the VHL protein, there was an inability to degrade the HIF alpha subunit under high oxygen conditions. And so, seizing on this clue, we and others rapidly showed that indeed the VHL complex is the ubiquitin ligase that targets the HIF alpha subunits for destruction in the presence of oxygen. And as we had predicted, VHL binds directly to the HIF alpha subunits through the beta domain. However, when oxygen levels are low or the VHL protein has been mutated, now HIF alpha can accumulate, dimerize with its partner protein ARNT, and activate various target genes such as VEGF. So this was very satisfying insofar as it explained why VHL defective tumors overproduce things such as VEGF and on occasion EPO. But it begged the question, how does the VHL protein know, if you will, whether oxygen is or is not present, and hence whether it should or should not destroy HIF alpha? So I'm going to share with you a couple of the experiments that were highly informative in this regard. So this is an experiment that was done by Haifeng Yang with TS20 mouse fibroblast. So these cells are VHL proficient, but they have a temperature-sensitive mutation in the E1 ubiquitin activating enzyme. So when these cells are shifted to the restrictive temperature, polyubicrylation will stop, and hence proteins that would have been earmarked for proteasomal degradation can accumulate. So in the bottom blot here, he did an anti-HIF1 alpha Western blot, and as expected, when the cells were shifted to the restrictive temperature, HIF1 alpha accumulated to high levels. But the truly informative experiment was the blot on the top. So in the blot on the top, rather than incubating the filter with an anti-HIF1 alpha antibody, he incubated the filter with the purified VHL alongin complex, and then after washing the filter, detected the bound VHL with an anti-VHL antibody. And here you could see that VHL could bind directly to HIF1 alpha, which wasn't frankly known at that time. But it could only do so if the HIF1 alpha had accumulated in the presence of oxygen. VHL could not recognize the HIF1 alpha that had accumulated in the absence of oxygen. And that told us something very important, that it was HIF1 alpha itself that was undergoing the putative oxygen-dependent modification that would earmark it for recognition by VHL and hence for destruction. It was also known at this time that iron chelators could mimic a hypoxic response. And although not shown here, Haifeng also showed that when you treated cells with iron chelators such as desferoxamine or iron antagonists such as cobalt chloride, again, HIF1 alpha would accumulate but was not recognized by the VHL protein. Now, Mircea Ivan in the lab then mapped the minimal region of HIF1 alpha that could bind to VHL. And he was able to narrow this down to a 25-mer HIF1 alpha peptide that at its core contained the sequence MLAP YIPM. So Mircea developed a VHL binding assay where he made this 25-mer peptide as a synthetic peptide with an N-terminal biotin that he could then capture on streptavidin and agarose and use to capture S35 labeled VHL protein made by in vitro translation. So he noticed that the HIF1 alpha peptide, this 25-mer, could capture the VHL protein, but only if it was first pre-incubated at 30 degrees with a mammalian cell extract, presumably to provide the necessary modification. Now, we were very lucky here because Jaime Caro at Thomas Jefferson had already been doing linker scanning mutagenesis of HIF1 alpha. And he had already shown that if you replace these 8 residues with 8 consecutive alanines, that rendered HIF1 alpha constitutively stable. And so Mircea made his 25 mer peptide, but now with these 8 residues converted to these 8 alanines, and as predicted based on the Jaime Caro result, now this peptide couldn't bind to VHL. So Mircea then did a so-called alanine scan, and he determined that one of the most important residues was this proline residue. So if he converted the proline to an alanine, now the HIF peptide could no longer bind to VHL. And so then we began to think about what potential modifications of proline could be potentially oxygen dependent and iron dependent. And we read more and more about the prolyl hydroxylases that, for example, modify collagen. And so Mircea now made his synthetic peptide with the proline already hydroxylated, and this peptide could capture VHL, but importantly, it no longer needed the pre-incubation step, presumably because the relevant modification had already taken place. Now, this said that the authentic modification was prolyl hydroxylation, or that prolyl hydroxylation could perhaps mimic the authentic modification, but we went on by mass spectrometry and other means to show that prolyl hydroxylation was the authentic modification in cells. And the same conclusion was reached in parallel by Peter Ratcliffe's group. So to summarize, in the presence of oxygen, HIF alpha becomes prolyl hydroxylated, and this generates a high affinity VHL binding site. Now, our group, using biochemical approaches, as well as Peter Ratcliffe and Stephen McKnight, using genetic approaches, identified the relevant prolyl hydroxylases as the so-called EGLN, or sometimes called PhD prolyl hydroxylases. So these enzymes split the molecular oxygen and use the oxygen atom to hydroxylate HIF alpha. Importantly, these enzymes, in contrast to the collagen prolyl hydroxylases, have very low oxygen affinities, and hence they're very sensitive to changes in oxygen in a physiologically relevant range. I'll also point out that these enzymes require iron, which explains the iron chelator or iron antagonist result. And finally, these enzymes require a cofactor, which is called variably 2-oxyglutarate or alpha ketoglutarate, which then gets decarboxylated to succinate during the hydroxylation reaction. Now, there are 3 members of the EGLN family: EGLN1, EGLN2, or EGLN3, with the alternative name shown here. But our work, as well as the work of Jacques Puysegur, clearly showed that the workhorse enzyme in terms of regulating HIF under normal circumstances is EGLN1. And perhaps for that reason, you can't make it through mammalian, or at least mouse embryogenesis, without EGLN1. So to circumvent this embryonic lethality, Andy Minamishima in the laboratory made mice where he could conditionally inactivate EGLN1 using an inducible Cree and a flox EGLN1 allele. So when he inactivated EGLN1 in adult mice, he could see that now the vessels were slightly engorged and the paws were slightly redder. And that's because these mice developed very significant polycythemia, or a very significant increase in red blood cell mass. Shortly thereafter, we learned from the work of Frank Lee and Joseph Percal and others that certain human families that develop polycythemia on a genetic basis have mutations in this very same pathway. So some families have homozygous, or less commonly compound heterozygous, hypomorphic VHL mutations, so-called Chuvash polycythemia. Yet other families have hypomorphic mutations in EGLN1, and yet other families have hypermorphic mutations in HIF2 alpha. So I think there's good genetic evidence in man and mouse that this is the oxygen sensing pathway, at least with respect to the control of red blood cell production. So in this mouse model I showed you a moment ago, the mice are making too many red blood cells, and that's because they're making too much EPO, and all of the EPO in these mice that's being overproduced is coming from the kidney. And that shouldn't be surprising, because in adult mammals, the kidney is the major source of EPO, but that's not true during fetal life. During fetal life, it's the liver which makes EPO, and then shortly after birth, the hepatic EPO locus becomes silenced, and the kidney takes over. And this has both medical and economic implications. So for example, in the United States, there are about 20 million Americans with chronic renal failure, and 2 to 4 million of these are anemic due at least partly to a relative insufficiency of EPO production by their kidneys. So Andy Minamishima wanted to know whether the hepatic EPO locus could be reactivated in adult mammals. And so he created mice where he inactivated the EGLN family members either singly in every pairwise combination, or he knocked out all three in the livers of adult mice. For comparison, he also knocked out VHL in the livers of adult mice. And what he saw was that when he knocked out all three family members, he saw a robust induction of hepatic EPO mRNA production, as shown in the black, as well as a significant increase in serum EPO, shown in the open bars. Now, in fairness, when he inactivated EGLN1 acutely, there was a pulse of EPO, but then that was dampened back down as you induced the other paralogs, which served sort of in a compensatory fashion. So you can generate either a pulse or sustained hepatic EPO production, depending on whether you knock out all three family members or you just target EGLN1 itself. Now, about 20 years ago, I reached out to a company called FibroGen, and I've already declared my conflict of interest. They were developing small molecule inhibitors of the collagen prolyl hydroxylases. These work in some cases by interfering with the utilization of that cofactor, alpha ketoglutarate. And I asked them whether we could test to see whether some of their collagen prolyl hydroxylase inhibitors might fortuitously inhibit the HIF prolyl hydroxylases, because if they did, we reasoned that would be a good starting point for medicinal chemistry efforts to make specific inhibitors of the HIF prolyl hydroxylases. So here's some data with one such tool compound from FibroGen, FG4497. This experiment was done by Ben Olenchuk, where he's treating cells in culture and then doing Western blots, and you can see nice induction of HIF1 alpha with this small molecule HIF prolyl hydroxylase inhibitor. Mikhail Safran in the lab had made mice that ubiquitously express a HIF1 alpha luciferase fusion protein, driven because the cDNA has been inserted into the ubiquitously expressed ROSA 26 locus. And now you can monitor HIP activation in these mice based on light emission. So here you're seeing mice that were given FG4497 by oral gavage, and you can see that in the mice given 4497, there's a stabilization of the HIP luciferase fusion protein, leading to increased photon emission. Now, ultimately, with medicinal chemistry efforts, this led to a first-in-class inhibitor of the HIF prolyl hydroxylases, this compound called roxadustat. Similar compounds have now been developed by several other pharmaceutical companies. Roxadustat has appeared to be helpful in phase 3 trials in patients with anemia linked to chronic kidney disease. Here are two of the early trials that were done in China. Roxadustat has now been approved for the treatment of anemia linked to chronic kidney disease in China and Japan, and the US FDA submission took place last December, and so we expect to hear whether it's been approved or not by the end of this year. Now, returning to the role of VHL in kidney cancer, so patients with VHL disease have one wild type VHL allele and one defective VHL allele, but initially they're okay because there's no evidence for haploinsufficiency of the VHL gene. As long as you have one wild type allele, you're okay. Over time, these patients can develop numerous pre-neoplastic renal cysts, and when examined, the cells lining these cyst cavities are VHL null. And then over years to decades, they can actually develop clear cell renal carcinomas. And when these tumors are examined, there are certain other stereotypical cooperating mutations in these tumors, in addition to loss of VHL itself. And so one conclusion is that inactivation of VHL is an important initiating or gatekeeper event for renal cell cancer, but it's not sufficient to cause renal cell carcinoma. Now, you could argue in fairness that the deck has been rigged here because, of course, the first event's going to be VHL loss, but what about in sporadic kidney cancers? Well, a similar picture emerges if you look at the work of Charlie Swanton and colleagues who have looked at sporadic clear cell renal cell carcinomas. and then have done multiple spatially distinct biopsies of those tumors, including in some cases, including metastatic deposits, and then have done used deep sequencing of those tumors and mutant allele frequencies to infer the evolutionary histories of these tumors. And almost invariably, they again see that inactivation of the VHL gene is the initiating or truncal event, and that there are again very similar stereotypical mutations to those seen in hereditary clear cell renal cell carcinoma that are required to take you all the way to a frank carcinoma. And some of these mutations occur in a subclonal or branching fashion, in contrast to the truncal mutation. So with all of these other mutations, it sort of begs the question, by the time you're a fully formed clear cell renal cell carcinoma, is VHL still relevant? Is HIF still relevant? And so to address this, many years ago, Othon and Leopoulos in the lab took VHL null renal cell carcinomas, and he restored the function of the VHL protein then by stable transfection, although we've subsequently done it by retroviral or lentiviral infection. Suffice it to say, when he restored the function of the VHL protein, those cells could still grow relatively happily on a plastic dish, but they lost the ability to form tumors in nude mice. And then to ask whether this was really about HIF, Keiichi Kondo in the lab took these cells and introduced into these cells a form of HIF alpha, and in particular HIF2 alpha, that can't be recognized by VHL because the proline hydroxylation site has been eliminated. And these cells regain the ability to form tumors. Conversely, when he eliminated HIF2 alpha in these cells using short hairpin RNA technology, or later we've done this with CRISPR, once again, these cells can grow on a plastic dish, but they lose the ability to form tumors in nude mice. And I should point out, importantly, this is a specific attribute of HIF2 alpha. The better studied, more famous member of the family, HIF1 alpha, doesn't do this. In fact, if anything, HIF1 alpha tends to suppress renal cell carcinoma growth. So the real bad guy here is HIF2 alpha. So what are we going to do about this therapeutically? Well, fortunately, by the late 90s, a number of companies were developing VEGF inhibitors. And we argued that if VEGF inhibitors were going to work anywhere in cancer, they should work in kidney cancer, where you have this intimate link between VHL loss, HIF activation, and subsequent induction of VEGF. In fact, here are data from Genentech, where they've measured VEGF production on the y-axis across a variety of tissue types on the x-axis. And in green are the VEGF levels in the normal tissues, and in red are the VEGF levels in the tumors arising from those tissues. And I think you can clearly see that kidney cancer is sort of the 800-pound gorilla, where VEGF levels go through the ceiling, presumably because from the get-go, these are tumors that are caused by VHL loss and chronic HIF activation. And we now have a number of VEGF inhibitors that have been approved for the treatment of kidney cancer. So that's the good news. And certainly some patients benefit from these drugs for a significant period of time. But some patients don't respond. And certainly the majority of patients who do respond will eventually develop resistance. So we need to do better. So I think you would argue, based on first principles, that rather than treating one HIF target gene, such as VEGF, maybe we should be targeting HIF itself. And based on what I just told you, HIF2 alpha. But unfortunately, classically, HIF2 alpha would have been viewed as being undruggable as a DNA binding transcription factor. But fortunately, Rick Bruick and Kevin Gardner, who were then at UT Southwestern, decided to ignore that dogma. And they identified a potentially druggable pocket in the so-called HIF2 alpha path B domain. And then at UT Southwestern, they identified chemicals that could bind to this pocket, and in so doing, induce an allosteric change in HIF2 alpha such that it can no longer dimerize with ARNT and hence no longer bind to DNA. So these chemicals were then out-licensed to a company called Peloton, and the scientists at Peloton did medicinal chemistry to make these chemicals more potent, more specific, and more bioavailable. And they were kind enough to share with us a compound that was closely related to their lead compound. So this tool compound was called PT2399. And Chin Cho in the lab, in preclinical experiments, showed that this chemical did everything you would want. It decreased HIF-dependent mRNAs when administered to renal carcinoma cell lines in cell culture. It prevented them from proliferating ex vivo, and it even suppressed their growth in orthotopic tumor models. So this was all very gratifying, but these are all so-called down assays, decreased transcription, decreased proliferation, and decreased tumor growth. And especially when you're doing a so-called down assay, you should be worried about so-called off-target effects, which is something I've written about in this recent review. So as a reminder, we frequently do experiments where we add some perturbant. It might be a drug, it might be an siRNA or a hairpin, it might be a CRISPR guide. It's been designed to inhibit some target, and then we measure some phenotype. But especially if it's a down phenotype that could just reflect a sick cell, a dying cell, a general loss of cellular fitness, we should always worry that we could be really looking at a so-called off-target effect. Now, geneticists have taught us for years that one way to deal with off-target effects is to do a rescue experiment. That is to design or discover a target that's going to be resistant to the perturbant, and now see if you reverse or block the phenotype. Now, of course, for siRNAs or CRISPR guides, this is pretty easy to do genetically. It's a little more difficult for a drug, but it can be done. And in this particular case, we were very fortunate that Bruick and Gardner had already identified a point mutation of HIF2 alpha that would prevent the HIF2 alpha inhibitor from binding to HIF2 alpha, and therefore would prevent it from inhibiting HIF2 alpha activity. And so Chin Cho in the lab used CRISPR to eliminate the endogenous HIF2 alpha in VHL null renal cell carcinoma cells, which you can do because these cells will tolerate loss of HIF2 alpha, at least in the short term, if grown under high serum. And she then took these cells and infected them with a lentivirus expressing either wild type HIF2 alpha or this putative drug resistant HIF2 alpha, this S304M mutant. So here by Western blot, I'm showing you using a HIF2 alpha antibody or a V5 antibody because the exogenous HIF2 alpha is V5 tag. You can see the exogenous HIF2 alpha expression here. NDRG1 is a well-studied HIF target gene, and you can see induction of this HIF2 target gene. And I should point out that if anything, the drug-resistant mutant is slightly enfeebled as a HIF in terms of its ability to activate HIF target genes. But that's good because if you're going to introduce a bias into your experiment, the bias should always work against you, right? So here the drug-resistant mutant, if anything, is a less potent HIF transcription vector. Okay, so now let's ask whether the ability of this drug to inhibit HIF responsive mRNAs is actually on target. So let's measure 4 different HIF2 target genes shown here by real-time PCR. So we're going to measure the mRNA levels of these 4 very well-studied HIF2 dependent genes. In red are the cells expressing the drug-resistant mutant, and you can see these cells are relatively resistant in contrast to the blue. The blue are the cells that are expressing wild-type HIF2 alpha. So from this experiment, we can say that at least the downregulation of HIF2 responsive mRNAs is on target because it's largely rescued by the drug-resistant HIF2 alpha. That was also the case in soft agar assay. So here I'm showing you a typical soft agar assay where we treated a VHL null renal carcinoma cell line with the HIF2 alpha inhibitor PT2399 at 0.2 or 2 micromolar, and you can see a decrease in soft agar growth. But in fairness, I could do this with Clorox bleach. I could do this with formalin if I titrated the dose properly. So do we really know this is on target? Well, we know this is on target because when Chin did this with isogenic cells that had the drug-resistant mutation, now these cells were insensitive to the drug in this assay. And finally, this was also true in tumor assays. So here, Chin has injected nude mice by tail vein with a VHL null renal carcinoma cell line obtained from Joan Massaquet that also expresses luciferase. So we can monitor the growth of these pulmonary nodules by bioluminescent imaging. So if we just focus on the cells treated with the HIF2 alpha inhibitor, here are the cells without the drug-resistant mutant, and here are the cells with the drug-resistant mutant. And so we could say that this effect is on target as well. Now, you might say that I'm cheating because we were handed on a silver platter a drug resistant mutant for our target of interest. What do you do if you're not quite so lucky? Well, an approach that we've used successfully was an approach used by Charles Sawyers and George Daly and others, where on occasion for other targets, we've taken an expression vector for the target of interest. We've grown them in error-prone E. coli, such as XL1 red that you can buy from Strategene. We've then taken this pool of mutagenized vector and introduced it into a sensitive cell line that was then treated with the drug of interest. And we've sequenced the plasmid DNA from the survivors to identify drug-resistant variants. And this has worked very well for us for a number of other targets. Okay, so this work was summarized together with similar work by Jim Bruguerola, showing that these HIF2 alpha antagonists were highly active in a subset of VHL null renal cell carcinomas. So based in part on these data, this HIF2 alpha inhibitor went into clinical testing. Here are recent data with the newest version of this HIF2 alpha inhibitor that's now recently been acquired by Merck. So these are so-called swimmer's plots. So each horizontal bar is an individual patient and how long they stayed on therapy. These patients had advanced kidney cancer and had failed multiple lines of prior therapy. Almost all of them had failed a VEGF inhibitor, and many of them had also failed a so-called immune checkpoint inhibitor. For orientation, this dotted line is 1 year on therapy. All of the patients with the black arrows at the time of this analysis were doing well and continuing on therapy. And the patients with the yellow stars are patients who had actual RECIST partial responses. So this is sort of a glass half full. Clearly, it looks like some of these patients are benefiting, but some of these patients are not, and we need to understand that better. Now, I should point out, again, these are patients who were heavily pre-treated, and so perhaps some of these patients would have responded to a HIF2 alpha inhibitor had they been treated in the front line rather than in the third, fourth, or fifth line. So to begin to address that, we were able to convince Peloton to do a trial in patients who had von Hippel-Lindau disease-associated renal cell carcinomas, taking advantage of the fact that many of these patients have small but measurable renal cell carcinomas that are just being monitored in careful surveillance programs in an attempt to avoid doing multiple surgeries and in an attempt to preserve renal function. And so a study was led by Eric Yonash that's going to be presented at ASCO this year. This is the publicly available abstract. And so there were 61 patients with VHL disease who had measurable renal cell carcinomas. Many of these patients also had other measurable tumors, including hemangioblastomas and paragangliomas. The vast majority of the patients demonstrated some measurable tumor shrinkage. About 28% have had confirmed partial responses at this time. Another 13% have partial responses that await independent confirmation. The median progression-free survival has not been reached, and the 12-month progression-free survival is 98.3%. And notably, responses have also been seen in some of their other tumors, including hemangioblastomas and tumors arising in their pancreases. Now, these are statistics just to end and give this sort of a human face. Some of these patients have started posting their results on Facebook. And so if you go on Facebook, you can find posts like these where patients are reporting to one another that they're responding. Here's a patient saying, "I never thought I'd see this day." Some patients are actually posting their pre and post scans. I wasn't sure I should do that, but here's an example of a patient describing how some of their tumors are now stable or getting smaller and in some cases disappearing. And then in closing, I was sent this vlog from a patient on the trial, and I thought it would be fun just to spend a couple.
Hey everybody, it's Justin. I just wanted to give you a quick update. I am in a gondola right now in Taiwan. Over there is Taipei 101. The gondola is actually right by the Taipei Zoo. But I just wanted to give you a quick update and say I'm doing well. I'm enjoying my trip. If it wasn't for the PT2977 drug trial, I would have never been able to come out here and do what I'm doing right now. So I just want to thank Peloton, and I hope Merck will fast track this drug for a VHL treatment. So if you guys are listening, hopefully you guys will put it on the market to help VHL. But yeah, keep watching these videos. I'll be making more, and I'll get better at it, and I have to get the angles right because I kind of look fat, you know? Anyways, I'll show you some of the views.
So anyway, I think that's great, because I think a young man like this should be worried more about whether his video makes his face look fat, and not worrying about whether his next doctor's visit is going to be associated with very bad news about his tumor. So hopefully this is an important step in the right direction to being able to manage this disease, as well as to make progress in sporadic kidney cancer. So thank you very much for your attention.
Bill, thank you for a wonderful keynote lecture. The questions are piling in. I think it was lovely to see Justin as well, and hopefully Mark will fast track that HIF2 alpha inhibitor. One of the questions is, it has been shown that cancer cells stressed with hypoxia downmodulate their DNA repair machinery, especially MMR, in a HIF-dependent manner. Can you comment about this particular HIF-driven activity, and does it affect the mutational burden in renal carcinoma?
Yeah, that's a great question. So by nature, I'm what's called a lumper. I try to lump things together, but sometimes you have to be a splitter. So what do we learn? We've learned that, first of all, HIF1 and HIF2 have some overlapping functions, but they have some distinct functions that are unique to either HIF1 or HIF2. And then if we look at the genes that are regulated by, for example, HIF1 or HIF2, the ones that get the most attention are the ones we think of as potentially promoting tumor growth, such as VEGF. But on further inspection, especially in the case of HIF1, it also regulates some genes that you would argue might suppress tumor growth or create certain liabilities. And so, for example, when you activate HIF1, you shut down protein translation through another protein called RED1. So all this to say is HIF biology gets pretty complex. And then as the question suggests, we are beginning to learn from the work of Celeste Simon and others that HIF clearly plays a role in a regulation of DNA damage. I'm not smart enough to tell you whether the mutational signature in kidney cancer is due to that HIF activation. I think it's a wonderful question. And then I think we're also interested in whether that creates certain therapeutic vulnerabilities. Although I will say in that regard that kidney cancer is amongst the most difficult cancers to treat with conventional cytotoxic chemotherapy. So apparently, whatever the impairment is in DNA repair, it doesn't seem to be making them highly susceptible to conventional chemotherapeutic agents. But thankfully, we now have newer drugs such as the HIF2 inhibitor that seem to be active.
So I think alongside going further along with that sort of discussion, can you comment on the cases in which HIF proteins appear to act as tumor suppressors instead of oncogenes?
Yeah, another great question. So if you look at publicly available gene dependency data sets, such as the DepMap at the Broad Institute, perhaps somewhat to people's surprise, you would find that most cancer cell lines representing many different lineages, actually, if anything, in those cells, HIF1 seems to be acting as a tumor suppressor. But a very big caveat, of course, there is that these experiments are usually done on a plastic dish in the presence of plenty of oxygen. So I think what we would find in vivo is still an open question. I think there are examples where it's clearly been demonstrated in at least mouse models that HIF1 can either promote tumor growth or suppress tumor growth in a context-dependent fashion. But this leads to the broader question I'm frequently asked, which is, if you had a HIF1 inhibitor, where would you use it? And I have to say, I'm not 100% sure. We don't really have that strong genetic smoking gun to say this is the patient population where you should test your HIF1 inhibitor. I can make some guesses, but as I just alluded to a moment ago, I think HIF biology is pretty complicated. And sometimes we get surprises when we actually do the right experiment in the mouse and we get rid of HIF1 and we actually see that the tumors get worse and not better.
And sort of following on from that, Bill, if a tumor is pseudo-hypoxic,
yes,
does it really matter what the dynamic microenvironmental hypoxia is like in terms of therapeutic attack?
Yeah, another great question. As the question relates to the fact that there are a number of tumors that have other mutations, such as, for example, in succinate dehydrogenase or fumarate hydratase, that again lead to upregulation of HIF, so-called pseudohypoxia. But I must say, in some of those tumors, it's not completely clear that HIF is the relevant target as opposed to other enzymes that potentially can sense both oxygen and alpha-ketoglutarate. So for example, there was a wonderful experiment done by the late Patrick Pollard, who was looking at fumarate hydratase lesions in mice that lacked fumarate hydratase. And I think many people in the field assumed this would be HIF. So when he co-deleted fumarate hydratase and HIF-1 alpha, the pathology got worse, in keeping with what I was just saying a moment ago. So we're still learning in some of these pseudohypoxic tumors what the cell intrinsic drivers are. I could still imagine that microenvironmental hypoxia might still be important, but certainly in those tumors, they're already, as you say, running that program, that hypoxic program.
Thanks, Bill. Oops, it's jumped a little bit. This is an interesting question. So we know Tibetans who have been adapted to living at high altitude have mutations in HIF2, confer an advantage in oxygen transportation without causing polycythemia. So do you know why this is the case?
Yeah, it's a great question, and it has multiple layers to it. So the first is, I'm sometimes asked, why do those familial polycythemia patients not look like VHL disease, but now we can also extend it to people living at high altitude? I think it's the same phenomenon. So whether you have one of these familial polycythemia syndromes or you're living at high altitude, you have a slight excess of HIF, but every cell in the body that could be making more EPO is now making slightly more EPO. That's in contrast to, for example, a kidney cancer where you have a clonal population of cells that's perhaps wildly overexpressing HIF, but the rest of the body is still normal or intact. So one possibility is just quantitative, like a little bit of HIF gets you polycythemia, and even more HIF above some threshold would finally get you to cancer. And there's some evidence to support that. But it could also be qualitative. So for example, we know that VHL has multiple other functions in addition to HIF that might contribute to tumorigenesis. Again, I used the word hypomorphic earlier when I was describing the VHL mutations seen in the Schubass polycythemia. These are really quite subtle mutations, but as I said, it's a subtle mutation multiplied by many, many cells, as opposed to the clonal situation with the cancer.
There are still questions pouring in. I don't know if we're going to get through them all, Bill, but can you just comment further on why some patients with clear cell carcinoma are resistant to the HIF2 inhibitor?
Yes, well, that's why I actually love this recent VHL trial that I closed with, because when we did the initial trial in the sporadic population, as I showed you, many patients responded, but many did not. And it left open the possibility that, some kidney cancers from the get-go are using a different pathway that we don't know about yet, that some of these VHL defective kidney cancers are perhaps never dependent on HIF2. An alternative explanation would have been maybe early on they were HIF2 dependent, but after you beat them up with multiple rounds of therapy, finally a clone emerged that was no longer dependent on HIF2. I think the VHL trial gives me some encouragement that if these drugs could be moved in the front line, a much higher percentage of the patients will respond and be HIF2 dependent, in which case this question becomes, I think, slightly less pressing. In other words, what are the other mechanisms that the tumor cells are using to become HIF2 independent?
Next question. Do you know how hypoxia may affect the formation of the so-called tumor dormant cells, which eventually affect disease relapse?
Yeah, I'm going to cheat here a little bit because the short answer is I don't completely know the answer, but I'll tell you what little I know, or at least we're thinking about. I think there's very good evidence that hypoxia, in addition to regulating the HIF pathway, also regulates a number of other enzymes, including some of the histone demethylases that we and others have shown are also oxygen sensitive. So I think this whole idea of a hypoxic niche and now a hypoxic tumor affecting cellular behavior through not only HIF, but through epigenetic reprogramming is starting to get some legs. And then secondly, I mentioned the difference between HIF-1 and HIF-2. HIF-1 in a lot of cells almost puts them in a state of hibernation, presumably so they can conserve ATP in a low oxygen environment. So that starts to smell a little bit like dormancy to me. So I think it's a great question.
And the next one is, a long time ago, medroxyprogesterone and tamoxifen were used to treat renal clear cell carcinoma with some success. So the question is, does HIF interact somehow with steroid hormone receptor and signaling?
Yeah, that's another great question, but I'm not going to put my medical oncologist hat back on that I usually don't do anymore. So it turns out kidney cancer is really quite notorious for the fact that almost anything you do, upwards of 5% of patients will respond. And in fact, in the early trials of the VEGF inhibitors, the treatment of kidney cancer was so bad you could justify having a placebo group that was treated with normal saline. And one of the patients treated with normal saline had a RECIST partial response. And that, frankly, was one of the reasons why people began to suspect that kidney cancer might be immunogenic, because you would occasionally see these spontaneous regressions in patients with kidney cancer. So I'm a little skeptical that the MEGACE and progesterone are really acting above noise. There's always a little bit of noise in any clinical trial with kidney cancer patients.
Okay, the next question, Bill, is really about, I think, drug delivery. So hypoxic tumor microenvironments are very poorly vascularized.
Yes.
So can this affect the HIF-targeted therapy strategy?
Yes, I think that's a very good question. I used to study tumor blood flow way back when I was in medical school. And it's true that tumors contain lots of regions that are hypoxic, but if you actually study them, some of the vessels are opening and closing, sludging and unsludging. And so over the course of time, a region that was hypoxic yesterday might not be hypoxic tomorrow. So it's actually pretty complicated. It would have been a concern going in, but the empiric observation is that the HIF-2 inhibitors seem to be working in these tumors linked to VHL loss. But I think this is a general concern with any tumor and any targeted agent, is whether you can deliver quite enough. I will say on the flip side that tumor vessels are notoriously leaky, and sometimes that can help you too, unless the interstitial pressure gets to be too high.
Bill, sort of sticking with that, bit controversial. I wonder what your views are on vasculogenic mimicry by tumor cells, and whether that's a potential resistance mechanism for either HIF or anti-angiogenic therapies.
I must say, I think the jury is out on the clinical significance of that fascinating phenomenon. I'll also lump pseudo-renormalization in that bucket too, of things that I think is intriguing, but I don't know the clinical relevance quite yet. You can always think of a reason why something might fail, and so yes, you might have imagined that the VEGF inhibitors and the HIF-2 inhibitors would be done in because of vascular mimicry, but so far, thankfully, it doesn't seem to be a major problem.
Okay, we're coming to the end of our time for the Q&A, Bill. It just leaves me to thank you for a wonderful keynote. I know you moved heaven and earth to be with us for the Q&A yes. today, and we really appreciate that you got with us.
Well, I still have my armband from the doctor, but I'm here. I made it.
Yeah, all right.
I got the IV taken out in time, so we're good.
Okay, thanks so much. It was great to see you.
Thank you.